Okay, welcome to what turned out to be part 2 of a discussion of protein structural classification levels. And I ended the other video right after the discussion of the E.F. Hand motif in secondary structure. So let me just summarize. Part 1 would include all the way to almost all the way there. So I'll give it a one-sided arrow there. So discussion of primary structure, mutations, secondary structure, alpha helices, beta sheets, and one motif. And now the second part of this video, second part of this topic, the video will include the discussion of the other motif and a discussion of tertiary structure, domains, and quaternary structure. Okay, so let's go ahead and get into it. Alrighty, so the second motif that we're going to talk about, that we're going to discuss, is called a beta-alpha-beta motif. This motif consists of two parallel beta strands that are connected to each other by an alpha helix. Okay, so you have one beta strand, and then coming out of the carboxy terminus of that beta strand, you have an alpha helix that weaves back around and connects to the amino terminus of the other beta strand, and then it proceeds on. So basically, remember, these two beta strands are hydrogen bonded to each other extensively. Okay, so you've got hydrogen bonds that exist between the backbone atoms of these two beta strands, and then coming above the plane of the sheet is this alpha helix that then comes back around and attaches to the other end of that next beta strand. Okay, when you put several of these together in a protein, that's very common. You make what's called a beta barrel motif, and it looks like a barrel. All the beta strands, usually there's 8 of them, I believe, and they're all facing the same direction, and you've got all these helices on the outside. Usually at the mouth of that resulting donut, you would have most of the activity there. Okay, so you would have active loops that come out there and do the business. Okay, so that is an alpha-beta-alpha motif. So now we'll start a discussion of tertiary structure. Tertiary structure is the next level. So we talked about primary structure, which is sequence, and secondary structure, which is localized, ordered arrangements of the protein backbone that's stabilized by hydrogen bonds between those backbone atoms. In tertiary structure, we are discussing the three-dimensional folding of the protein. Okay, so it's at this point that you would most likely begin to think about actual structure if you didn't know any different. This is what you would think of as folding. Okay, now tertiary structure is stabilized by hydrogen bonds, dipole interactions, salt bridges, dispersion forces. So all the possible intermolecular interactions that can exist between the R group, so between the side chains of the amino acids that make up the protein, and also between the backbone atoms, so the backbone amide atoms there can interact with R groups or with each other. So, but the type of interactions that they're going to participate in, whether they're backbone or side chain, are going to be the common intermolecular interactions that we've already talked about. Okay. In addition to this, you've got one other kind of stabilizing presence, and it's not an intermolecular interaction. It is actually a covalent bond that can be formed between two cysteine residues. So you can put two cysteine residues together, and you can oxidize those, and it becomes a disulfide, what's called a disulfide bond. That bond is a covalent bond, so it is very, very strong. Okay, so it's the strongest type of interaction that can stabilize tertiary structure. And here I've kind of drawn a schematic of it. So you've got a cysteine, and this can be a cysteine in another subunit. It's not as likely to do that, to get that inner subunit disulfide, but sometimes you can, or it can be in the same protein chain. Just wraps around, goes crazy, comes back around. And these two cysteines are placed in such a way that the disulfide bond can be introduced here. So the oxidation happens, and you end up with this covalent bond between those two sulfur atoms. And that is what is called a disulfide bond. It is, as I said, very strong. And it's used to stabilize tertiary structure, so that once the protein folds correctly, these bonds can be introduced, and they can help hold the protein the way it's supposed to be. Okay, now we're going to talk about some common characteristics of tertiary structure that you might not have thought of before. And to begin, I'm going to tell you that in tertiary structure, amino acids that are very far apart in sequence are brought close to each other in space. So I want you to think about that, because when you fold a protein, kind of like beta strands can be far apart in sequence, a three-dimensionally folded protein that is folded up on itself is going to have pieces of that protein that are very far apart from each other in actual sequence, that are brought very, very close in space. So don't let that confuse you when we get into the deeper stuff and we talk about protein sequence and folding, and numbering and stuff like that. Okay. The next thing is that when proteins fold, they want to put their hydrophobic side chains. So the hydrophobic residues want to be buried inside the protein, and they do this to escape from water. And you'll remember this, of course, because this is the entropic force that drives the release of that water, is the entropic force that drives protein folding in the first place. So the idea that hydrophobic side chains tend to be buried on the inside of a protein, and hydrophilic residues don't mind being on the outside. In fact, they like it because they can form interactions with water. And that tends to be a very stable place for them to be. Okay. Now, related to that is this idea. Water is almost always excluded from the interior of a protein. Okay, so once it's folded three-dimensionally, think about a big glob. It's very, very structured, though, highly complex. Water is not found in the interior of that protein unless it is there for a very, very specific reason. Okay, so if it's there to help to make the structure form correctly, or if it's there to be catalytically active, which is usually the case if you've got a water on the inside of a protein on the interior folded in there. It's usually a catalytic water. Otherwise, you're not going to have water on the interior of a protein at all. And this causes something that's kind of interesting to happen. So if you think about two charges that would be attracted to each other on the surface of a protein, or before the protein folds, you have here a negatively charged acidic residue and a basic residue. The attraction between these two side chains is actually going to be extraordinarily reduced when the side chains are exposed to aqueous solvents. So water molecules are actually going to associate with these charges. Of course, they're going to be oriented oppositely. I don't even know if that's a word, but anyway, you know what I mean. In a way that, of course, with the negative charge, the hydrogens of the water would be pointing toward that negatively charged residue, and here the oxygens would be reversed, pointing toward this positively charged residue. But either way, the charges are shielded by these shells of water, and that makes the attractive force between them actually reduced a great deal. And so you want to think about that when you think about these guys being on the surface of a protein. Okay. However, water is excluded from the interior of a protein. So if you have a salt bridge or a hydrogen bond or even dipole interactions on the interior of a protein where there is no water, those salt bridges and hydrogen bonds are extra strong on the interior of that protein, way stronger than they would be on the surface exposed to solvent. Okay, and that's because you don't have the shielding waters. You don't have those water shells that would be stabilizing the charges and blocking interaction with the oppositely charged residue. Okay, so just remember that these guys may seem to be fairly weak when they're on the surface and exposed to water, but on the inside they're much stronger. Okay. All right, so now we're going to talk about a special example of tertiary structure. So motifs are secondary structure arrangements. Domains are tertiary structure related structural things. Okay, so that was technical term there, structural things. Okay, domains are independently folded regions of the same protein chain. That's very important. Okay, so when you have a protein that is greater than, say, about 200 amino acids, you begin to be able to form these domains. And that is where part of the protein chain folds independently of the other part, and you end up with these two areas that are joined to each other in sequence, but separate as far as their folding goes. Usually they're joined by some sort of loop region that separates them. And so here are two examples, and I'm going to give you these examples that will also include some of the different functions that domains can play. Usually you're not going to form domains unless you need to have different functions for those domains, okay? So in this case I drew this protein with two domains, and you can see that because they're separately folded, parts of the same chain, one amino end, one carboxy end. This end could be, say, a regulatory domain. The regulatory domain would be responsible then for controlling the activity of the catalytic domain. So the regulatory domain could bind in some way to the catalytic domain and turn it on, or it could bind and turn that catalytic domain off. Very often it will be like bound, it'll be over here bound up next to the catalytic domain, blocking the active site, and then when some effector molecule binds to that regulatory domain, it would cause a conformational change that would allow the regulatory domain to float, away, still tethered too, of course, because it's all one chain, but it would then allow the catalytic domain either to be turned on or turned off, depending on the type of system you got going on. Okay. You also have transcription factors. Transcription factors have two domains. So again, we have one amino end and one carboxy end here. So this is all one protein chain. But these proteins don't do catalysis. They're not enzymes. Their job is to bind to promoter regions of genes and activate transcription of a gene. Okay? So if you need to make a protein, then a transcription factor very often will come down and bind to the promoter region that's right before the beginning of the instructions for making the protein. And so you'll have a DNA binding domain, so part of the protein that's independently folded will bind to and recognize a specific sequence of DNA that's in that promoter region, and it'll bind there. And then the other domain serves as a beacon, and its job would be to call and tell RNA polymerase, which is an enzyme that that actually transcribes DNA into RNA. It would serve as a beacon to it to come and make a transcript of this gene and to go ahead and make the protein. Okay? All right, so this is an example of two different proteins that have completely different roles. This protein would be some sort of enzyme with its catalytic activity and its regulatory domain. Okay, this protein has no catalytic activity. Its job is to act as a transcription factor, so it has a domain that binds to specific sequence of DNA in a promoter region of a gene and signals for transcription of that gene to happen with this domain up here, this N-terminus domain. Okay, so that is the discussion of domains. And now we are going to finish up, ta-da, with our last level of protein structure, and that is quaternary structure. Quaternary structure is actually pretty straightforward. There's not a lot to say about it right now. We'll talk about it when I give you the details in lecture. Quaternary structure involves the assembly of subunits. Okay, so you cannot have quaternary structure if you don't have more than one subunit. That would be stupid. You can't assemble subunits if you only have one. So quaternary structure describes the interactions between the subunits. So remember, with hemoglobin, we had 2 alpha and 2 beta subunits. So we have quaternary structure in hemoglobin. Here in white, you have the interactions between the subunits, the intermolecular interactions between those subunits. That is the quaternary structure, the assembly of these subunits, how they're oriented with respect to each other. And remember, they're really not this way in hemoglobin. They are tetrahedral, kind of in their arrangement with respect to each other. So all these positions are equivalent. Okay, so quaternary structure involves the same stabilizing forces as tertiary structure does. So hydrogen bonds, dipoles, salt bridges, dispersion forces, but the presence of disulfides is much less common in quaternary structure. It's not very common to have subunits that are covalently linked to each other through disulfide bonds. Okay? All right, so that is quaternary structure. I'm trying to think if there's anything else I need to tell you. We're going to talk about, in lecture, I'll give you specific examples how each of these levels is important. Okay? And so we'll talk more detail about that and more about protein structure later in lecture. So I appreciate your attention, and I hope you learned a lot, and I hope you don't have to listen to this too many times because that could be annoying for you. But as far as I'm concerned, this is the end of this topic. Woohoo!